Hi, awesome. Thank you so much, Philip, for the great introduction. Hi, everyone. Thank you so much for joining me for my talk today on the radiation impact on the sarcoma immune microenvironment. My name is Chang Su, and I'm a MD PhD student in Dr. David Kirsch's lab at Duke University. So as you all know, that immunotherapy has now become a standard cancer therapy for many patients. When it first came out, it received very dramatic response rate, especially in melanoma patients, close to 25%. However, majority of the patients do not respond just to anti-PD-1 reagents alone. And we kind of saw the same trend in sarcoma patients. Here's a figure from SARC 28, where 93% of the 40 patients that was enrolled in the study that had unresectable or metastatic soft tissue sarcoma had progressive disease even after receiving pembrolizumab. And so we were wondering if we combined it with radiation therapy with anti-PD-1, maybe we would see better response because radiation therapy has been shown to kind of promote immunogenicity and sometimes even elicit an abscopal effect. So this is a mouse study that we conducted a study in, which is p53 MCA autochthonous sarcoma model. It's a combined genetically engineered and carcinogen-induced mouse model. So we actually induce a tumor in wild-type mouse, and then we inject the gastrocnemius site with adenovirus that contains CRISPR-Cas9 and single-guided RNA to p53. So that knocks out p53 in the muscle cells. And then we also follow up with an injection of MCA, which is a carcinogen, and that induced mutations in the gastrocnemius muscle. In about 6 to 10 weeks, tumor develops at the muscle site, and the histology pathology is very similar to undifferentiated pleomorphic sarcoma in human. And the reason that we picked the p53 MCA model compared to KP, like KRAS being turned on and p53 being knocked out, so a pure genetically engineered mouse model, is because KP model has only about a median of 18 non-synonymous somatic mutations. So it's not quite strong enough to form enough kind of mutate proteins for the immune system to mount an attack to. And then for p53 MCA model, we have a median about 2004 mutations. So we think it's a better model for studying immunotherapy. And then so we conducted the study, and on the left figure here, you can see that autochthonous p53 MCA tumor, if we don't give it any treatment, which is the solid blue lines, the tumor grows very quickly, and then when we give it anti-PD-1 alone, it didn't respond, super well, as was predicted by the clinical trial. However, what's, I guess, more surprising is that when we give anti-PD-1 with 20 gray of radiation, it didn't seem like it did anything compared to if we just give it radiation alone. However, we took these tumors and we dissociated them and generated a cell line. And then we injected it into syngeneic mouse. So this is a common kind of transplant tumor model. But in this model, we actually saw a dramatic response. If we give 20 anti-PD-1 and 20 gray of radiation, 100% of the mice actually gets cured from their tumors. So this kind of tells us that p53 MCA model mimics kind of the coevolution of tumor cells as well as the immune system as in the human cancer would. And transplant models have an immune environment that seems to be more conducive for immunotherapy. And then so p53 MCA probably has better kind of clinical predictive values in terms of therapy. And so we were thinking what other immunotherapies that might work better in the sarcoma model, since anti-PD-1 doesn't seem to be working as well. And CpG, which is a TLR9 agonist, kind of caught our eyes because Dr. Levy's lab from Stanford published a paper where they gave CpG and anti-OX40 to mouse with primary mammary gland tumors, and they received a really good response. And so what is CpG? It is a TLR9 agonist, which can activate plasmacytoid dendritic cells, as well as B cells, and then mostly plasmacytoid dendritic cells to secrete interferon alpha, which then can activate NK cells to kill tumor cells. It also helps plasmacytoid dendritic cells to mature and migrate to the lymph nodes and express CD80, CD86, CD40 co-stimulatory molecules to cross-present antigens to the T cells and activate them for cytotoxic killing of tumor cells. This is our kind of experimental design, which is on day 0, I initiate the tumor. In about 7 to 11 weeks, the tumor becomes measurable at 70 to 150 cubic millimeters. And then on that day, we give it either 0 gray or 20 gray of radiation to the mouse at a gastrocnemius muscle site. And then on day 3, the mouse receives CpG or CpG control. And then a week later, we repeat the CpG or CpG control therapy. And on the right here, you can see this is time to tumor quintupling. So the days it takes for the tumor to quintuple in size, and this was monitored by 3 times a week caliber measurement. And you can see that on the left there, that's the control group that didn't receive any kind of therapy. And within like 7 to 10 days, tumor all quintuples in size. However, on the far right, that's the CpG plus radiation therapy group. And you can see that it takes much longer, like a month or even sometimes close to 2 months for the tumor to actually quintuple in size. And so we wanted to see what immune cell population might be responsible for such treatment effect by CpG plus radiation therapy. So this time, I induced the tumor on day 0. However, I waited till the tumor is a little bit bigger to 180 to 300 cubic millimeters before I initiated the treatment, so we'll have enough tumor tissues to harvest, essentially, for the following experiment. And on day 0, we give 0 gray or 20 gray of radiation. After 3 days, we give CpG or CpG control. And on day 6, we harvest the tumor. And we perform tumor dissociation with enzymes, and then we perform sorting with FACS for live cells without debris, because debris have a huge impact kind of on the mass cytometry machine. And then we label the cells with antibodies that's conjugated to heavy metals. And as you can see, so this is unbiased clustering of kind of all this, the CD45 positive cells. And you can see at the right lower quadrant, that's treated with CpG plus radiation therapy and has more CD3 T cells compared to the other groups. And here again, CpG plus radiation therapy group on the right bottom quadrant has significantly more CD8 T cells when compared to radiation therapy alone, CpG alone, or the control group that didn't receive any treatment. This is granzyme B expression, which is a good marker to indicate that these CD8 T cells are active in tumor killing instead of being exhausted or it's not in an active state. And CpG plus radiation therapy, again, has more T cells that's expressing granzyme B, high levels of granzyme B. And this is kind of histology to confirm that. So this is CD8 T cell staining within for the tumor tissues. And as you can see, radiation, so the little brown dots are CD8 T cells. You just have a lot more of them in the RT plus CpG group compared to non-treated group or the radiation therapy or CPG treatment alone. So for the second experiment, we wanted to see what are the kind of gene and pathway expression profiles for these CD8 T cells. So we did a single-cell RNA sequencing. The tumors are harvested the same way at the same time points, except this time we sorted out only CD45 positive cells. So only the immune cells got sequenced. And here is a UMAP for kind of all the immune cells. And as you can see, majority of the cells are actually monocytes and macrophages. And we did a subclustering of just the T cells and NK cells. And as you can see from kind of the left figure here, that the CD8 T cells, which are shown in blue, you have significantly more of them in the radiation therapy plus CPG group. Again, compared to radiation therapy alone or CPG alone or tumor with no treatment. And we looked at granzyme B expression within the CD8 T cells, which are circled in red. And you also have more granzyme B expressions in the radiation therapy plus CPG group, which confirms what we saw on CyTOF. And it actually also have much higher interferon gamma expression as well in the CD8 T cells after treatment with radiation therapy plus CPG. And so after that experiment, we also performed bulk RNA sequencing because in the single cell, we only looked at immune cells. And so we wanted to look at tumor cell expression. Like what markers are being expressed by tumor cells as well. So the tumors are still treated and harvested at the same time points. And this time we saved half of the tumor for histology and the other half for RNA extraction. And the histology staining was what you saw earlier, was the CyTOF data. And this is analyzed by Dr. Molding at Stanford using CyberSort. And as you can see that on the left figure, the fourth column is the CPG plus radiation therapy group. It has the kind of the highest, almost 80% of them are immune cells. And we think this might be due to that the cancer cells were actively dying and they're probably less resistant to the enzyme treatment process, and that's thus resulting in like almost 80% of the cells being immune cells. And you can see that there's a significant increase in CD8 T cells, as well as activated NK cells in the CPG plus radiation therapy group. So this was a paper that was published in Nature where the group use the TCGA data as well as other data set to kind of classify sarcomas into 5 different groups due to their like immune compositions. And group D and E, so class D and E on the far right, are associated with better overall clinical survival. And I think group E is also associated with better response rate to pembrolizumab. And so we actually clustered our tumor cells, mouse tumor cells, according to their algorithm as well. And as you can see, the purple group kind of on top, which is the radiation therapy plus CPG, most of them cluster to group B and E, meaning that they are more likely to be associated with better survival and response rate to pembrolizumab. And this is a quick kind of to explain the data. We looked at TCR clonal expansion, and this is a toy example for a score that's called the evenness score. So essentially, if you have 1 or 2 clones that really expanded versus all the T cells that really expanded, you will have a lower evenness score versus if you have 4 different types of T cells and they all expanded equally, then you still have an evenness score of 1. And what that is to say is that in our CPG plus radiation therapy group, the evenness score is much lower, meaning that only certain T cell clones expanded compared to all of them. And that probably means that only the kind of tumor antigen-specific T cells likely expanded after CpG plus radiation therapy. And we confirmed this finding kind of in vivo, where we use CD8 anti-CD8 antibodies to deplete CD8 T cells. So on the left is just with CD8 isotype antibody. So CD8 intact group mimics what we saw earlier. However, in the CD8 depleted groups, the CpG treatment alone effect, as well as CpG plus radiation therapy treatment effect, goes away. And so future opportunities in sarcoma patients, personally, we think CpG has really great potentials for clinical trial, because one, has really great safety profiles. There are several ongoing clinical trials with just CpG or in CpG with combination of some other therapies. And also we can actually do this as the intratumoral injection, which even further reduces kind of the toxicity possibility in patients. And this is actually a study done by Dr. Monjazab at UC Davis. And this is spontaneous canine tumors, where the first, so figure B there is a canine model with melanoma in its oral cavity. And then figure D is actually a sarcoma in the dog, companion dogs. And they were treated with radiation therapy plus CpG weekly for 4 weeks. And as you can see, not only does the primary tumor goes away, on the right figure, those are actually metastases to the lung. They also shrink as well. So there's almost an abscopal effect. So in summary, CpG plus radiation therapy significantly reduced tumor growth rate. CD8 T cells increase with CpG plus radiation at RNA and at protein levels. Depletion of CD8 T cells really negate the anti-tumor effects of CpG plus radiation therapy. Clonal expansion of T cells that are likely tumor antigen specific, and CpG can be administered as the intratumoral injections. And with that, I would like to thank Dr. David Kirsch for his mentorship and everyone else that helped me with the project. And I will take any questions.